Today I'll be going to talk about the computational tool that we have developed that can predict type 9 secretion system and associated functionalities. I'll start my presentation by saying that protein secretion is very much a fundamental and essential biological process that is conserved across all forms of life. And if we focus on prokaryotes and in particular bacteria, and we see that the bacteria actually have developed protein complexes, basically protein machinery, which actually facilitate the secretion of proteins. And in case of gram-positive bacteria, where you have one membrane, the secretion is actually pretty much very simple, and this is mediated by export pathways. And in case of gram-negative bacteria, there are dedicated secretion systems, and these protein complexes are present in the membranes, and they can, with the help of export pathways, secrete proteins to the extracellular space and even to the host cell membrane. So there are some secretion system which can directly take the protein from the cell to extracellular space, and also there are secretion system which can take the protein from periplasmic region here to the extracellular space, and some can directly take the protein from cell to host cell membrane. So there are 11 such secretion system got so far discovered, and I will be focusing on type 9 secretion system, which is in short T9SS. So I would like to give a brief introduction about what is type 9 secretion system, what is it, why it is important. So type 9 secretion system was initially discovered in Porphyromonas gingivalis, which is a periodontal pathogen in 2005, and subsequently the naming came in 2013. And there are several experiments which has been published, and where people have observed the presence of the secretion system in other pathogens as well. And also, so far the experiments and bioinformatics analysis suggest that the secretion system is exclusive to Bacteroidetes phylum, which is a very diverse phylum which is present in different microbiota. And there is a particular type of motility which is associated with the secretion system. So I'm going to show a video. So I would like your attention in this bacteria, which is a rod-shaped bacteria, which is Flabobacterium johnsoniae, which is found in soil microbiota. And this dot-shaped structure that you see, these are the proteins which are secreted by this bacteria using type 9 secretion system, and they stick to the surface of the bacteria and helps in the motility. The important point here is that these bacteria do not have flagella or pili. Using this secretion system, they can move on the surface. So this particular phenomenon is called gliding motility, and this bacteria uses this gliding motility to nutrient uptake and survival. And there is another important feature is that there are bacteria where T9SS is present, but those bacteria are also non-motile. They do not exhibit gliding motility. And one such example is Porphyromonas gingivalis, which is a periodontal pathogen, but that do not have motility, but some has motility. And also there are some bacteria, for example, Johnsoniae, which actually secretes digestive enzyme which can actually degrade complex carbohydrates. And for biotechnology application perspective also, the secretion system is very much important. So what is the question that we are interested in is the following. So initially what we did is, this is a plot of the published literature versus different secretion system. And what we see is that type 9 secretion system is one of the least characterized secretion system, and it is a very much recently discovered. So, and there is not much of literature. And prior to our work, there was not a dedicated resource which can actually predict the type 9 secretion system and all its associated functionalities. So what is the question that we are asking is that we are interested in these three questions. Can we develop a tool which can actually predict the presence of type 9 secretion system and gliding motility and secreted protein in a bacteria where this particular secretion system is not characterized experimentally? So to do this, first what we did is we looked at the literature, published literature, and we found more than 100 such literature. And we manually curated data from this literature, and we finally compiled 23 proteins where there are experimental evidence of them being associated with T9SS in bacteria. and 22 proteins for gliding motility, and we have also compiled 102 secreted proteins. And we see that there is a large overlap between the proteins which are associated with T9SS and gliding motility, which suggests that there is a interlinking between T9SS and gliding motility, which also people have previously observed. And this is a schematics that we have drawn based on the subcellular localization of this protein based on the literature, and to understand how exactly the secretion happens. So not much is known about all the components. Very few components has been experimentally characterized. So these proteins which are synthesized in the cell, so these have N-terminal signal peptide and C-terminal domain, and it requires the set export pathway for the transcript of this protein to the periplasm. So for that, the signal peptide is required, and once it is in the periplasmic region, then there are these proteins which are highlighted. These proteins plays a very important role according to their function, which is reported in the literature, and they interact with the C-terminal domain and allows the transport of this protein through this SPRA, which is a big beta-barrel membrane protein, through the outer membrane. And then the further post-translational modification takes place. So from this visualization we sort of, and from the literature we understood that these are the 6 proteins which are very much functionally important for the secretion via T9SS. So next question that we are asking, which is important for us to develop a tool, that is, are there any protein which are actually minimal set of protein which we can say mandatory for the T9SS based secretion or gliding motility? So to answer that question, what we did is we take 30 experimentally characterized bacteroidetes, and these bacteroidetes can be classified into mainly 2 groups. So one is like where you have the experimental evidence for presence and absence of T9SS, and then presence and absence of gliding motility. So, and then we see the presence of this protein across these 30 bacteroidetes, and then this helps us making a case which are the mandatory proteins for the T9SS and gliding motility. So these are the 6 bacteroidetes where T9SS is experimentally characterized, versus these 2 where T9SS, these 2 bacteroidetes do not have T9SS. And we see that these 6 protein which is functionally important, they are always present in these 6 bacteroidetes which having T9SS. Similarly, if you take these 22 bacteroidetes where gliding motility is observed versus these 8 bacteroidetes where they do not have gliding motility, we see that additional 5, total 11 proteins actually important, which we call mandatory component. So what we did is, this is the rule that we are going to follow for the prediction. That is, given a bacterial proteome, we check for the presence of 6 T9SS mandatory component. If these proteins are present, we call them that these bacteroidetes can have T9SS. And additionally, in addition to the mandatory T9SS mandatory component, we check for another 5. If all these 11 proteins are present, we say that this bacteria has gliding motility, can exhibit gliding motility. So using this, we have a simple Python program which we call T9GPRED. That is the tool we have developed, which is openly accessible. I will be talking about that in the last slide. And using that tool, we have predicted the presence of T9SS across 693 completely sequenced bacteroidetes strains which is available. And we see that 402 strains have T9SS, and out of 402, 327 strains are predicted to have also gliding motility. Further, we have also checked the presence of T9SS across bacterial kingdom. So we have considered more than 34,000 bacterial sequence, and we see that T9SS is only predicted only in Bacteroidetes phylum, which is also observed by various other groups previously. And further, we have also taken 4 newly characterized T9SS bacteria where T9SS is characterized, which was not covered in our model building. And we see that our tool is able to predict all of them to have T9SS. Similarly, we have also taken 7 new bacteria where gliding motility is observed, and we see that our tool is able to predict all 7 to have gliding motility. So next, we also have observed from the literature that some of this mandatory component, which we have considered 6 mandatory component, they forms operon-like cluster in their genome. So what we did is we sort of observed, this is just a visualization we did. So these are the two model bacteroidetes where T9SS and gliding motility actually studied. So we see that these four proteins actually form operon. This is an experimental fact. So what we have done is we asked the question that we had the more than 400 bacteroidetes where we predict the presence of T9SS, and we have checked whether they also form sort of cluster in their genome based on the location of the gene. And we see that almost all of these bacteroidetes where we predict the presence of T9SS, these four protein also forms clustering. So that would be important for some application perspective. Now I'll be going to the secreted protein prediction. So as I was explaining, the C-terminal domain of the protein, secreted protein, acts as the secretion signal. The question is, what are these CTD? Are there any classification of this CTD? So what we did is, and there was no such clear understanding of what exactly are the CTD, what are the classes. So what we did is we sort of compiled 18 sequence motifs from literature which is published, and we did there are ME suite. Using that you can actually compare different motifs also, and we compared these motifs and found that there are two such kind of motifs which exist in for CTD, and there is type A and type B, and there is also another motifs which are very rare for type 9 based secretion system, the secreted protein, which is type C, which is found in Kia type domain. So what we have done is we have taken all these motifs and available HMM models which are available for these CTDs, and we have classified the 102 secreted protein that we have compiled for this different kind of CTD, and we are able to classify some for type A, some for type B, and there was one protein for type C, and there are some protein which we are not able to classify. Maybe they may have some novel CTD which experimentally is not characterized. So further we have the pipeline where we given a protein sequence, we check for the if it is experimentally annotated to be a secreted protein, we call that as a secreted protein. If not, then we check for all these features, and that is it should have a signal peptide because type 9 secretion system requires set export pathway, and then we check for other features, and finally we check for the presence of C-terminal domain. So if it has any of these three CTD types, then we say that it is a protein secreted via T9SS. Further, we have also validated our result with small test dataset which was available. So there was 22 newly available secreted protein which were experimentally characterized in 4 different strains, and we see that majority of them we are able to predict as secreted protein. We have randomized the sequence of this C-terminal domain, and we see and estimated the precision of our tool, and we see that it is high. And lastly, we ask one important question, that whether this C-terminal domain is unique to type 9 based secretion or it can be found in other secreted protein. So bacterial as I was saying, it is a very diverse phylum. It does not have only T9SS, but it can have T1SS, T4, T5, T6. So what we have done is we have taken the secreted protein from all the secretion system and checked the presence of the C-terminal domain in all this secreted protein, and we find that the CTD is very much unique to T9SS based secretion system and not found in secreted protein via the secretion system. So here we have found some interesting result here. So what we have done here is that we have compared the secreted protein for gliding bacteroidetes that we have predicted versus bacteroidetes where we have predicted that it is not gliding, and we found that the gliding bacteroidetes seems to have secreting more protein than non-gliding bacteroidetes. And for the genome size also we checked, and we also see that it is also significantly larger than the non-gliding counterparts. So the hypothesis that we have is that it seems like gliding bacteroidetes seems to have significantly larger genome sizes, and it probably encode more secreted protein in comparison to non-gliding bacteroidetes. And finally, we have made this web survey where this is available in this URL, and it has browse section where one can browse the prediction and details for 693 completely sequence bacteroidetes. And further, we have this prediction pills where one can input the sequence, bacterial protein sequence, and it can give you the presence and presence of T9SS gliding motility, and also if a protein can be secreted also, it can tell you. And this tool is actually, the web server is limited, have limited computational power, but if you want to actually use it locally, then the corresponding GitHub repository is available where one can actually download all the codes and run it locally. So, so finally, I'd like to acknowledge my advisor, Professor Arjit Samal, and my co-authors, and I am SCH DA. Thanks.
Interesting talk. So, any questions?
Very good presentation. Two queries. One is that your prediction is based on that 9, you have mentioned some 9 proteins previously. Yes, 6 protein. Okay,
yes.
6 protein.
Yes.
Why are you considering all these 6 should be mandatory?
So, so these...
Because these are present in your data set.
So, all the experimentally validated bacteroidetes
Yes.
where T9SS is present, these 6 are always present.
I am sharing one experience of my group.
Okay.
We are working on the exosome.
Okay.
And when you look for the exosome which are present in throughout the life, yes, what it is showing that from the bacterial kingdom to the archaeal kingdom to the it's completely changed. Even within the bacterial kingdom, some domain has lost, some is not present. And there is maybe the different combinations of the proteins which made with the same kind of functions. If you have to say these are the mandatory proteins, then you have to delete any one of them experimentally, and so the secretion function has been lost. Otherwise, probably this will be the overstatement.
Okay.
Yeah, this is.
So actually, there are actually experiments where suggest that these 4 proteins that you see KLMN, they forms a complex, and so this complex actually drives the secretion. And if you mutate, or if one of the protein is absent, then the secretion cannot happen. Now, for the interaction with the CTD, this SPRase protein is really important. So this has been observed in this model organism, and this SPRA protein is actually the beta-viral protein which actually allows the transport through that protein to the outer membrane. Now, there are actually paper which also does similar sort of thing, and they also take mandatory proteins and says that these are the mandatory proteins based on one can predict different secretion system, and we have also followed similar sort of.
Yes, but into account your argument. But my point is that until and unless you delete any one of them and show it experimentally, it lost the secretion property. Probably in my logic, you can't say these are the mandatory proteins. In maximum, you can say these are observed in most of all the species which has these particular.
So for that, actually, we did this simple sort of comparison.
I agree with. He didn't understand the question. So the question is, you have collected data from papers, right? Have any of the papers characterized the structure of the protein, or have the mutation and shown that secretion is...
These are the only protein where it is characterized experimentally properly. That is, you have the structures. And
the question is, have the paper done a mutation of any of these proteins and shown that secretion is lost?
So other proteins also there are mutation, but these proteins are, when they do mutation, they are complete loss of the secretion.
So you started with experimental data to begin with for your prediction.
Yes. Yes.
Okay. So there's a question in the...
Quick 2 questions. So one is the follow-up of this question. So how these 6 sequence of proteins are conserved across all these bacteroides?
So these are the experimental set where we see that they are conserved. But if you see bacteroidetes, as I was saying, so these are so we have taken 693 bacteroidetes, and we see that all these 6 proteins are conserved across these 402 strains.
With certain identity or something like that, right?
Sorry?
With certain identity, percentage of identity of these sequences.
So what we have done, this prediction that we are doing based on HMM profiles, which we have built. So what we have done is we have taken the sequence and aligned them and trimmed the unconserved region, and only taken the domain which is conserved. So they are actually domain-wise, they are actually conserved.
And then a quick second question, is there any relation of virulence with this whole T9SS pathways or something.
So, yes. So, for example, Porphyromonas gingivalis, which is a dental pathogen that uses T9SS and secrete enzymes which actually destroys our dental tissue. So, there are also other bacteria which are actually pathogens to poultry, birds, and fish, which can actually actually secrete virulent
So
pathogens.
it's directly associated with pathogenicity that you.
There are some bacteria where T9SS is associated with virulence, some where it is not.
Okay, there's one last, some more questions here.
Yes, you guys know the structure of these proteins known.
So some of them are actually known. For example, these mandatory components actually that I have showed, these structures are actually known. For other components, there is no structure so far.
Did you check alpha fold?
So yes, sir. So for the secreted protein, what we have done is whatever prediction we have done, we have annotated them with Uniprot ID or AlphaFold ID wherever it is available. So we have.
Last question in the interest of time.
It's a very nice overview of this work. I'm just wondering, is there a relationship between different secretion systems?
Okay, so there we have not actually done that, but there are paper where it tries to sort of see these components where it is present in other secretion system, and they have not found any homologous of the secretion system in a bacteria where T9SS is not present. So this is how actually it was discovered initially in gingivalis. They are trying to see if any other secretion system which is already known present in that, but they did not find it, and that is how they started finding different components which are present in gingivalis, and the secretion system actually came into picture.
Thank you, Ajay, for an interesting piece of work.